So last but not least, Cliff Tabin from Harvard Medical School will tell us about the signals and the forces that pattern the intestinal smooth muscle.
Okay, thank you very much. So I'm going to be telling you, as the title says, a story about the morphogenesis of the gut, and in particular the morphogenesis of the smooth muscle of the gut. Now, although I'm going to be talking about the gut, I want to just sort of set this up just conceptually by looking at a picture of a limb. And here you have the tricep and the bicep. Now, if I were to ask you by their name, why does the bicep have 2 heads and the tricep have 3 heads? No one knows. Why is the bicep even there? Why do you have a muscle in that location? Nobody knows. Okay, let's stipulate that you have these muscles. Why do the fibers run this way instead of this way or in some random way? Again, for all that we know about limb development, which is a very intensively studied system, for all that we know about muscles, muscle stem cells, muscle specification genes, if we don't know the answer to those questions, I'd say that we still don't really understand muscle development or limb development very well. So what I'd like to do is try and answer those questions, how you get muscles in the right place and how you get the fibers in the right orientation. But I'm going to try and tell you this story in a different context, a simpler context, and that of the developing gut. So this is from a human embryology textbook. My talk is almost all chick. There's one mouse slide in there, I think. But the anatomy would be the same. You have the endoderm with the villi on the inside, and around it are a couple of different layers of muscle. The inner layer of muscle here is circumferential, that is, the fibers are pointing this way. And the next layer is longitudinal, and the outer layer, they're running along this length. And those 2 layers are important for peristalsis. You all know that. As your food moves through the gut, the circumferential muscles squeeze behind. You have a relaxation of the longitudinal muscles in front, and the food moves down. Now, these muscle layers develop over time sequentially. So it's very importantly for the story I'm going to tell, and I'm going to tell 2 halves to the story, but for both halves, it turns out to be important that the first layer that forms is this one here, the inner layer, the circumferential layer, and the second layer is the longitudinal layer. You can see that here. This is at an early stage, doesn't matter. It happens to be embryonic day 6 in the chick, but early stage you have the circumferential layer, you don't have the outer layer. Subsequently, you get the second layer here. So we want to know, how do you get the spatial and temporal control of the patterning? And I will tell you now that that's going to turn out to be a signaling story, not a physical forces story. And then how do you get the orientation of the fibers in the right orientation? That turns out to be based on physical forces. So we're going to start out with the first one. How do you get the muscle layers in the right place? These muscle layers are organized sort of like a target in sort of circumferential concentric circles. When you think of concentric organization as a developmental biologist, one thinks of radial signals. For example, in the fly or in the butterfly, you have the wing pattern of the fly imaginal disc, where you have, and this is an oversimplified, fly people, please don't jump on me, this is an oversimplified version of the story, but essentially you have a signal in the center, wingless, that it leads in a sort of like a morphogen, different distances from the source of the signal, you get different stripes. Well, we have a signal on the inside of the gut. The endoderm of the gut produces, and we've known since 1995, when my former postdoc Drusilla Roberts was looking at hedgehog in the chick, that the endoderm of the gut expresses sonic hedgehog. This is a gene called patched. For the people in the room who aren't biologists, what it is doesn't really matter, other than it's a readout for cells that respond to hedgehog. So it forms a gradient. And then you can find other genes that come on with different threshold levels of hedgehog at different distances from the endoderm that produces. And I should say the endoderm itself does not respond to hedgehog. There's no patched in the endoderm. So the endoderm makes hedgehog and doesn't make it. All right, so an obvious sort of idea then would be that maybe in some graded fashion, you might have hedgehog inducing the formation of these muscle layers. The problem with this idea is that the literature is contradictory. A couple different, because hedgehog's been very well studied. Hedgehog's been knocked out. The pathway's been knocked out by knocking out Smoothen by Andy McMahon. Both Indian and sonic hedgehog are produced the same way in the midgut, the intestine. They've both been knocked out by Deb Gamuccio. If you get rid of hedgehog signaling, you lose the muscle. You increase hedgehog signaling, get more muscle. The mouse data says hedgehog induces muscle. But other groups have published in the chick that in chick explants, If you add an inhibitor of hedgehog, you actually, by inhibiting hedgehog, get more muscle. And if you add a lot of hedgehog to the culture system, by increasing hedgehog, you lose muscle. So hedgehog inhibits muscle. Now that seems confusing. It could be a species difference, which doesn't really feel right. But sort of an obvious solution to this would be that they're both actually correct and that you've got a morphogen type response. One could imagine anyway that hedgehog is indeed necessary to make muscle, but high levels of hedgehog inhibit muscle. If you knock it out completely in the mouse, you lose it, but if you overexpress it too high in the chick, you inhibit it. So a graduate student of mine, Tyler Huck, decided to try and test this model, and he did it by chick explants. We can grow guts from a stage where there is no muscle to a stage where that muscle layer is formed. If you do that, you do get the muscle right where it should be. This is just a readout of hedgehog signaling. This is again patched, but this is the smooth muscle and smooth muscle actin. And as the mouse data would have said, hedgehog does seem to be upstream of muscle because if you block hedgehog signaling with an inhibitor called cyclopamine, no hedgehog signaling, no muscle. A little bit of hedgehog signaling, a little bit of muscle. More hedgehog signaling, more muscle. So the mouse model seems to be right. But if you add enough hedgehog signaling, you lose muscle. So enough hedgehog signaling, you block it. So it does seem like there's this sort of response where at the high levels you're blocking, low levels you're inducing. But one could imagine that this is in fact direct, that hedgehog has two different effects on muscle at different concentrations, but we could also imagine that it's indirect, that hedgehog has an effect on muscle, which is to induce it, but it also induces something else at a high concentration, and that something else is what's inhibiting. And leading us towards at least considering that model is that one of these genes I showed you before, a high threshold response to hedgehog, is another secreted signal, BMP4. And indeed, you can show that in this context that hedgehog is upstream of BMP4. So there's BMP4 in the wild type directly under the endoderm. You block hedgehog signaling, you get no BMP4. You have a huge amount of hedgehog signaling, everybody expresses BMP4. So hedgehog is indeed upstream of BMP4. Is BMP4 in fact inhibitory of the smooth muscle? So here's the normal smooth muscle and the control. And you add BMP4 and you completely lose smooth muscle. Now that looks a lot like what happens if you add huge amounts of hedgehog. With huge amounts of hedgehog, you also have the absence of muscle. We're arguing now that that's because a lot of hedgehogs inducing a lot of BMP4. If that were true, you add the hedgehog and block BMP4, the prediction would be in the absence of BMP4, now the muscle should form. And you can block BMP with an inhibitor noggin. And if you do that, the same high level of hedgehog gives you not an absence of muscle, but everything turns to muscle. So we think that this is the right model for what's happening in terms of getting this gap. This BMP4 is the reason that you don't have muscle right up against the endoderm. But you also don't have the muscle forming out here where the later layers of muscle are going to form. And we think the reason that there's nothing forming out here is because independent of hedgehog signaling, there's constitutive expression of two other members of the BMP family, BMP2 and BMP7, on the outside of the developing gut. So that gives you BMPs on both sides, which leaves you with sort of a sweet spot in the middle where you have low BMP signaling, where the hedgehog signaling that's necessary can give you that first layer. All right, so it's a simple signaling story of how you get that first layer, but now how are you going to get the second layer where you have BMPs inhibiting it? Well, one way you could think that you could get rid of it would be if you had an inhibitor endogenously knocking down BMP. And indeed, an endogenous inhibitor, noggin, is expressed by that first muscle layer as soon as it forms. So as the first layer forms, it starts inhibiting the gradient of BMP. Now you don't want the next layer to form both on the inside and the outside. You only want it to form on the outside. And you add to that level of noggin a second expression of noggin, which is the enteric nervous system. As the enteric neurons migrate in from the outside to innervate the forming muscle layer, it also expresses noggin. So now you have noggin both in the muscle and adjacent to the muscle, giving you a knockdown of that BMP gradient. One might expect that that would lead to the ability for now hedgehog to induce muscle further out. And indeed, if you add a lot of noggin to the system early on, before this occurs, So you can get both muscle layers to form at the same time. So if you add noggin early on, now not only do you have the inner layer forming, you have the outer layer forming at a stage when in the control, the outer layer hasn't yet formed. This gap in here is not cells that aren't forming noggin. These are the enteric nervous system cells. So basically, you're turning everything to muscle. If you put in noggin early on, you can turn everything to noggin, everything to muscle. And these cells here are the neural crest enteric neurons. Well, that says noggin can do it. Is noggin actually doing it? So noggin's been knocked out in the mouse. So we contacted Richard Harlan, who knocked out noggin, and said, can you send us, well, first we just asked him, what's the smooth muscle look like in the gut in the noggin knockout? He said, well, it certainly has smooth muscle. It looks like it's fine. But of course, if you don't look in section carefully at the orientation and the layers, it would look like it had muscle. So we asked him if he could actually send us the mice. This is the control. So it's just showing you that in a mouse, you have the same layers as you do in the chick. There's the inner layer and the outer layer at E16.5. And the reason I'm looking at E16.5 and not later is because the noggin mutant dies at 16.5. But that's late enough to see the two layers. And if you get the noggin mutant, that outer layer is cleanly lost. There's no effect on that first forming layer, but you never get the outer layer if you can't make noggin. We wanted to look later than 16.5, so we also did it in a conditional way, using a pair of different crees that will remove a flox noggin allele from both the neural crest derived enteric nervous system and from the smooth muscle. And if you lose it, you lose almost all of the muscle. There's a little bit still there that comes on in spite of the fact that you've lost noggin. And we think that might have to do with redundancy because I haven't shown you, but the cells that express noggin also express chordin and gremlin, two other inhibitors of BMP. So we think that we have a sense of how you get the layers where they belong. You start out with the circumferential layer forming in the sweet spot where hedgehog signaling can form in the absence of BMP signaling. Then you have this gradient of noggin that comes on that pushes back the BMP gradient on the outside, allowing another layer to form. So now I want to switch gears, and I want to talk about a second problem, and that is how do you get the orientation of the fibers? So, and this is just to show you that the fibers are in different orientations. So far I've been showing you things in section, where you sort of looking at it from the side, but you can also instead image with a 2-photon microscope from the side at different depths to look at the orientation of the fibers. And if you do that looking at the orientation of the fibers at an early stage, when there's only one layer, you can see that they're oriented, this direction is circumferential. And at a later stage, the inner layer is circumferential, and the outer layer is forming, and it's forming perpendicular to it in a longitudinal direction. So the first smooth layer has its layers oriented circumferentially. What would be causing the layers to orient in that particular orientation? And a cue to that came from people who work and do tissue engineering. So tissue engineers for many, many years have done different things to manipulate cells in vitro. And there's a long published literature that both fibroblasts, myofibroblasts, and vascular smooth muscle cells, if you provide a stretch, orient in the direction of the stretch. So if you plate smooth muscle cells on a rubber sheet and then just pull on them, the cells will line up in the direction that they're stretched. And this is a subset of the publications. There are many more than that. So if you had a situation where the tissue itself was being stretched, that would give you an orientation that the cells might be able to use. And of course, as you have a tube that's being pushed outward as it's growing, you get this circumferential tension in the wall of the tube, very much like the stretch that you have on the surface of a balloon as it's being blown up. And it's very easy to show that there is such a stretch, which we think is coming because as the muscle cells drop out of if everything was growing at the same rate, you wouldn't have a stretch. But what happens is as the smooth muscle cells start to differentiate in random orientations, they lower the rate of cell division. Yet the tissue on the inside is still proliferating at a high rate. So it's pushing outward on the tissue because you have more growth on the inside than you have on the outside. And it's easy to show that simply by making a cut. So this is, there it is. So this is a donut of the tube, if you want. And you can see here, here is the muscle that's differentiating. Here's the tissue on the inside. And if you cut right here, separating the inner stuff from the muscle, the inner stuff, mesenchyme and endoderm, expand outward, meaning that it was constrained, it was compressed. And meanwhile, the muscle tissue, as soon as you remove this, expands inward, meaning that it was strained. And once you have the barrier removed, it collapses inward. And as I said, we think the reason that this is occurring is because of the growth of this tissue being greater than the growth of this tissue. So one way to test that would be to drive high level of growth in all the cells. So you can misexpress various oncogenes or various growth factors that will drive high and uniform proliferation throughout the tissue. So if you have uniform tissue, so instead of having, this is just a cartoon of what I just said, if this is normally growing faster, so it's compressed, and this is growing slower, so it's being stretched. But if you make the tissue at the same rate, you should be able to get rid of that stretch. One way of measuring the stretch is by residual strain. So if you cut the tube, make a slit on the side, it'll pop open to relieve the strain, right? And the degree to which it pops open is a measure of how much strain there was. So if you use either c-Myc or myristoylated AKT, you can drive the expression of these two genes through a retrovirus. You get uniform proliferation throughout the tissue, which I'm not showing you, but you can measure that by BRDU incorporation. And with uniform proliferation, you get rid of almost the entirety of the strain. So this is the normal strain, and this is the degree to which this pops open relative to the degree that it pops open in the wild type. And the result of that is, if the strain isn't there, the cells orient in random orientations. And that's shown in pictures and also quantified here. Now, of course, you could argue in this experiment that the reason these cells are randomized is not because of strain, but because you're misexpressing Myc, and Myc has some other effect on the cells. So another way to do the experiment is to relieve the strain physically. I already showed you that you can relieve the strain by making a cut in it. So you remove the tension. Now you can culture this without the cut, in which case you get the oriented fibers, which we've already shown, or culture it flat without strain. And if you culture it flat without, so these are the two culture systems. Either way, you get the muscle layer forming. In the control, the fibers are all oriented correctly. But if you do have it flat, the fibers are randomized. And you can again quantify that. And instead of having well-oriented fibers at 90 degrees, which means circumferentially, you have basically no orientation to the fibers. Now, if that's the case, if it's really just stretch that's doing it, you should be able to put this on one of those machines that stretches things, one of these machines, like here, and stretch it. And if you stretch it, the argument would be that if you stretch it, you should be able to reapply the stretch, you should be able to give it the orientation. And that's indeed what happens. So this is culturing flat, and if you culture flat by itself, without stretching it, the cells are random. But if you stretch it, now they achieve an orientation based on whatever direction you stretch it. So we think we know something about how you get that first orientation. The first orientation is that you have the cells in here proliferating, putting a strain in the cells on the outside of them, and in response to that strain, the cells align in the parallel to the stretch. And that gives you the circumferential muscle layer. So is the same sort of thing going on for the second layer? That is to say, if you culture it flat, will the second layer also be randomized? So in this case, we're going to cut it open flat after the first layer is already formed and say, now what happens to the second layer? Well, if you do that, the second layer forms with perfectly normal or longitudinal orientation. So the second layer is basing its orientation on something else. And it makes sense that it's basing it on something else because it's got to be in a different direction. So you've got that first layer that's circumferential because it's being pushed outward. You've got constant strain. What's making the second layer form perpendicularly? And what's the cue? And for that, we went back to the tissue engineers because they have other data that's also well established in the literature that if you do that same experiment, you plate the cells on a rubber sheet, vascular smooth muscle or fibroblasts, and you don't stretch them, but you stretch them cyclically. Stretch, relax, stretch, relax. You do that, and they orient perpendicular to the stretch. Now, that sounds kind of weird. I think the way, and it hasn't been worked out what the reason is, but I think what makes the most sense to me is that it's not really the cyclic stretching that's doing it, it's the cyclic compression. That they're stretched, they collapse in, you unstretch, stretch, unstretch them. Well, when it's stretched, the cells all line parallel along the direction of the, want to align along the line of the stretch, but then you compress them, and they don't like that. And so to try and minimize that compression on themselves, they reorient themselves perpendicularly. And of course, there is a cyclic compression going on in the developing gut. As soon as the gut forms, before the enteric nervous system gets there, the cells start contracting. The muscle fibers are circumferential, and with a much greater degree of in and out than you have from the pressure from the growth, you have this rippling effect from the first layer of smooth muscle. And the first layer of smooth muscle, or the smooth muscle contractions, are driven by calcium influxes. And this is just a picture just showing you calcium transients. So it's just showing you with a stain for a dye that shows you calcium transients. But the importance of that is just experimentally, there are lots of ways you can block calcium signaling. And we've done this with a number of different drugs. One of them is nifedipine. And I can show you this movie, although there's really nothing to see because this basically is rippling just like the wild type, and the one on the bottom isn't doing anything. So with nifedipine, you lose these cyclic contractions. So you add nifedipine, and instead of having the cells all aligned longitudinally, you lose the orientation. So now that's at 0 degrees, but now in this case, you've lost the orientation. And of course, that has no effect on the circumferential muscles that have already formed. Once they've formed and aligned, whether you block calcium signaling or not, calcium signaling had nothing to do with the orientation of that first layer. You can also go in the other direction. There are drugs that will increase the amount of calcium signaling. You'll see the increase, and this one's like bouncing out of the dish because it's contracting so much over here. And this is the wild type. And if you do that, then the cells hyper-orient themselves with this drug. And again, you can quantify that. And if you quantify it, you can see that this is with no calcium signaling, wild type, and with hypercalcium signaling or hypercontractions. Well, if these periodic contractions are what's orienting it, again, we can go back and test it with this machine. So you can take this machine and say, okay, let's block the second layer's orientation with nifedipine. So the first layer forms circumferentially, but you don't let that first layer contract because you add nifedipine. So now that first layer isn't contracting, you should get randomized cell orientation. But now you're going to go back in on top of that and put back in cyclic contractions. And when you do that, so here's normal development, nifedipine. Now you can't stretch the normal way. Squeezing it the way it normally goes is hard to do. So what we're going to do is put cyclic contractions in the longitudinal direction, so it actually orients them, gives them an orientation back through the cyclic contractions, but it's perpendicular to the normal one. So we think we have some understanding of what's going on here. The first layer is orienting based on strain from the static strains that are produced by the proliferation of the inner material, so both the endoderm and the mesenchyme, giving the cells aligning in a circumferential direction. Then as that first layer starts contracting, as they're contracting in a periodic manner, therefore as the next layer forms, the cells want to avoid the contraction of that, and they align perpendicular to it. And you can finally, the last little experiment I'll show you here, is that you can combine this knowledge to make an inside-out gut if you want. So what we're going to do now is, at the first stage, just put a static stretch on it, pulling longitudinally, so you're pulling harder this way than the growth is pushing outward this way. So if the major strain the cells feel is this way, you're going to try and orient them perpendicular at the first stage to the way they normally form. And that works. So that if you pull on them now, they're oriented longitudinally. The first layers are longitudinal instead of circumferential. Now both layers have formed. Of course, they both start contracting. The normal layer contracts normally, but the other layer contracts lengthwise. And because it's contracting lengthwise, the cells orient perpendicular to that contraction. So now the inner layer, instead of being circumferential, is longitudinal, and the outer layer, instead of being longitudinal, is circumferential. So we think we understand how you make these muscle layers, this anatomy that we talked about, at least in broad terms. There are a lot of details, obviously, we want to work out, but the timing and concentric pattern comes from this gradient of hedgehog being an inducer, BMP signaling, and the cascade where the first layer inhibits it secretes noggin, allowing another layer to form. And then these mechanical control of the fiber orientation of the second layer. And, in both cases, the sequential specification is necessary to get the layers to form correctly and one after another and to have different orientations. And the very last thing I just want to mention, I don't have time to show you any of the experiments, but there are lots of tubes in our bodies. And those tubes, a lot of them have smooth muscle. And it's worth noting that every tube that only has a single layer of smooth muscle, such as the blood vessels, that smooth muscle is circumferential. And there are lots of them that have more than one layer. For example, in the urogenital system, every tube that has two layers, the first layer is circumferential, the second one is longitudinal. And we've shown that in the ureter, I don't have time to show you, but a rotation student repeated some of these experiments in the ureter, and the ureter is exactly the same as the gut. So I think not the signaling of specifying the tissue layers is different from tube to tube, but the orienting of the layers within the tubes seems to be a general property of these developing systems. So again, Tyler Hike is the graduate student in my lab who did almost all the work. Bess Miller was the rotation student I just mentioned a couple seconds ago. Nanda Nurikar, who's now at Columbia, has worked on physical forces in the gut. We just published a paper that is more quantitative than this story on cell movements within the developing hindgut. And he helped sort of advise Tyler and think about this in a physical level, as did Mahadevan, who's a long-term collaborator of ours, as well as of Olivier's, who you heard about in the last talk. So I'll stop there, and I'll be glad to take questions.